note that I'm using QR codes for my presentation, so feel free to take photos that I can go back to during the question period if you like. All right, so let's get into it. So across the world and even in space, there exist extreme environments that typically forbid life from even forming. So these include parameters like extreme temperature, extremely cold to extremely hot, as well as acid and alkali, and salinity and radiation, to name a few among a variety of parameters that I didn't include here, but there's dozens of them. That being said, a subset of organisms that span all domains of life, bacteria, archaea, and eukaryotes, can actually adapt to survive and thrive in these conditions, and they're known as extremophiles. My presentation is really just going to be focusing on those adapted to extreme temperature and extreme pH, but we are starting to include other parameters in our investigations. So these extreme environments can impose strong selective pressures with both direct and indirect effects of the genome composition of species adapted to them. So starting with the psychrophiles, these are cold adapted species. DNA is quite stable in the cold, so we know it can go in the freezer for a long period of time and be okay. But in a living cell, it actually needs protein adaptations to be able to kind of keep that cellular integrity. So across all domains, pretty much all, if not most, if not all, psychrophiles have evolved these proteins known as antifreeze and isonucleating proteins, which can reduce and promote the growth of ice crystals, respectively. These have different impacts on cellular integrity. Moving on to the extreme temperature, those thermophiles and hyperthermophiles here, we know that DNA can actually be degraded by high temperatures. So these extreme conditions can denature our DNA and makes it quite unstable in these environments. Additionally, their high temperatures has an effect on proteins, seeing that high temperatures can actually break down the sort of integrity and therefore function of proteins, such as DNA polymerase, which would introduce mutations. So they evolve thermostable enzymes, which we're all familiar with from PCR. Moving on to the pH adapted extremophiles, starting with the acidophiles. These can actually undergo spontaneous reactions in acidic environments known as acidic depurination. So that's the loss of adenine and guanine. This is an effect that's pretty profound on its own, but it kind of exacerbated even more when you think of the strong bonding between G's and C's that hold the DNA together in this extremely harsh environment where it really needs that integrity. Going to the alkalophiles, so the extremely high pH alkaline environments, these can similarly undergo spontaneous reactions, taking away essentially hydroxide ions that breaks apart the phosphodiester bonding of the DNA backbone, breaking that backbone apart. So both these conditions can impact genome composition and integrity itself. All right, so overall, extremophiles use very versatile mechanisms to live, from the nucleic acid compositional level to the amino acid compositional level to make sure that those proteins can stay functional and hold their integrity. And here I'm just including an example of some of those proteins. So I mentioned those antifreeze proteins, which are found across a lot, if not most, of psychrophiles, as well as thermostable DNA polymerases that are in those thermophiles. Therefore, our central hypothesis asks whether extreme environments impose direct and indirect effects on the genome composition, such that organisms adapted to them have a characteristic genome-wide composition associated with environment. Not too sure what happened with my graphs here, but tables, but it did include two conditions for the acidic, so acidophiles and alkalophiles. And then for our temperature, I included psychrophiles, that's cold adapted, mesophiles, which is actually just normal conditions, 20 to 45 degrees Celsius, and then thermophiles and hyperthermophiles. So I believe thermophiles goes 45 to 80 degrees Celsius, hyperthermophiles is anything over, including over hundreds of boiling temperature, that can still survive, which is pretty cool. All right, so we investigated our extremophiles using genomic signatures. So genomic signatures are essentially unique patterns of oligonucleotides, also known as K-mers, that are pervasive across the genome. These patterns of oligonucleotides are shaped by pressures such as mutagenesis, selection, and population dynamics, making the genomic signature for a given species very, very specific and unique. Thus, they can be used to classify species by their kind of genomic signature identities. We can visualize these genomic signatures with a technique known as chaos game representation, which is sort of in this middle row here. So this is essentially a plot with specific areas where the oligonucleotide will go. So as you sort of feed in the collective composition of those oligonucleotides of the genome, we would begin plotting them according to the grid space. So in the red, as you can see kind of there, that actually corresponds to CTG, or sorry, CGT, sorry. So you would start at the middle, and you move a half distance to the vertex that represents that nucleotide in your oligo. So we would go half distance to our C verse, then to the G, and then to the T, landing us in that grid space. So we would do that as we go from the beginning to the end of the genome and get that visual representation of those Kmer counts. While this is very detailed, it's quite difficult for us to read, so we can create a frequency chaos gain representation plot, or FCGR. So that essentially overlays a grid on top of that sort of dot plot distribution. And instead of looking at those dots, it just kind of counts how many are there and then shades that grid space along a gradient. So the lighter a grid space is going to be, the less of that Kmer is frequent in the genome. The more that Kmer is frequent in the genome, the darker that grid space is going to be. We combine this genomic signature comparison with machine learning techniques. So our data set we curated with just under 700 bacteria Archaea extreme-file genomes, composing all of those different categories that I mentioned. We performed tenfold cross-validation, which is a sort of testing and training iterative technique to really fine-tune those details of the signature. We did both unsupervised and supervised machine learning, so we'll start with the supervised here at the top. Basically, the difference is supervised machine learning is going to take our data in conjunction with the labels and learn those patterns. So we tell it, okay, here's the genome, here's the environmental label and the taxonomic labels, and learn a pattern associated with them. So we did just that, and we found characteristic FCGRs that are associated with each environmental condition. So once we kind of have those signatures, the average signature of that environmental condition, we can actually take new DNA and predict what environment it would belong to, which is very cool and sort of kind of a proof that the environment does have whole genome compositional impacts, regardless of taxonomy and regardless of which domain, because we included both bacteria and Archaea. Our unsupervised machine learning is somewhat similar, but we essentially take away all the labels. So we just give our algorithms the data, and it needs to learn patterns and associate those patterns in the genomic signature data alone with no labeling. So it doesn't know what the taxonomy it is, it doesn't know what the environment is, it just wants to bring groups together based on similarity. There, we found a number of clusters, which are quite interesting, but the most interesting and even shocking was the clustering of bacteria with Archaea. So we had a couple of these groups where we had inter-domain clustering, which almost never happens considering the genetic distinctness between species of these two domains. We wanted to investigate that a little bit further because we only done like one or two and wanted to see if we can get more. So we played around with our parameters for our AI, playing around with the Kmer size, so changing the size of the oligo that we're looking at the DNA with. And then also the fragment size, so we're pulling different fragment lengths, trying to find that sweet spot for both computational speed and accuracy. After that sort of optimization, we discovered some putative pairs. I believe, yeah, there's 78 pairs, so quite a lot there. We wanted to filter them based on that FCGR similarity to make sure that the genomic signature composition really is quite similar to one another in the clustering. After which, we looked at them with environmental parameters to kind of really understand and deep dive into those clusters to understand why they're kind of put together more. This is a collection of the 5 groups that we have so far, and even through more parameter optimization, we get more clusters, which is very cool. So all of these clusters contain one bacteria paired with at least one Archaea. And I've included a legend here to kind of show the environmental labeling, where it's pretty much the same for all of the groups. So in group 1, we have thermophiles and acidophiles. Group 2, we have hyperthermophiles. Group 3, all hyperthermophiles as well. So it's kind of evidence to show that, okay, they are adapted to the same environment. Groups 4 and 5 I included also. I'm not showing much of those results because just for space reasons and time. But they do, it's worth noting, have different environmental conditions between the species. In our group 4, we actually have thermophilic bacteria clustering with mesophilic Archaea, and similar kind of idea with group 5 with one of those Archaea there being a thermophile, which is quite interesting still, trying to ask the question, why is something that's adapted to extreme heat clustering with something adapted to normal conditions? I just wanted to show this also because this is kind of a comment we got from our reviewers about how this convergence might actually be due to horizontal gene transfer, which is very present in extreme environments. But this sort of pairwise blast analysis, the alignment between the bacteria bacteria and archaea across the groups showed a query cover as low as 1% and as high as 2%, so barely any of those genomes are actually aligning together. Of those alignments, very moderate percentage identity, and the genes themselves that are aligning correspond to housekeeping genes, so non-adaptive proteins that wouldn't explain the environmental clustering. All right, so we actually pulled out from our genomic signatures between our different pairs here to sort of see what k-mers are actually important for environmental clustering. We then took those k-mers between our bacteria and archaea, and we performed Spearman rank correlation analysis to kind of see the correlation between those compositions, where a score of 1 is essentially perfect correlation. There we got row values of 0.96, so nearly perfect correlation for group 1, 0.81 for group 2, 0.79 for group 3, and very high values as well for group 4 and 5, demonstrating that these k-mers are in fact correlated in their compositions. Right, then we wanted to kind of look at the k-mers and those important k-mers for environmental classification in the context of codon usage bias, which is heavily documented in the sort of extremophile literature and sort of understanding what makes these genes work in extreme environments. So then we kind of cross-reference our 3-mers that are important, where we found that lots of them are matching. These are kind of similar to almost like a mutational signature plot, where we can see the trinucleotide, and then the green bars would be representing those important k-mers for classification. And they're pretty well near the same for a lot of our groups, even though one is bacteria and one is archaea. Again, almost never see that. When we compared it to the literature, we saw quite a bit of matching with our k-mers and the reported codons that have increased or decreased usage in the specific environments. So for example, group 1, we compared our species 3-mers to thermophiles and acidophile codon usage bias in the literature. We found lots of matching between those groups there, with yet to come because we're still getting more information as we keep going on these thermophilic genes and just broadly extremophilic genes. All right, then we wanted to see, it was pretty cool that our machine learning kind of brought together the bacteria and archaea computationally, but does it actually occur in the real world? So we tested this using a tool known as the Microbe Atlas Project. It's a great resource that compiles 16S rRNA and different metagenome reads from across the world in environmental samples. And so we looked up all of the environmental sample reads for each of our species and cross-referenced them to see, which projects, which environments are they both in at the same time. And we found lots of great results for that. So group 1, where we had the thermoacidophiles, we actually found them in the Washburn Hot Springs, which is in Yellowstone National Park, which happens to be a thermophilic, acidophilic environment. Group 2, we had a couple of co-occurrences, as we term them, also composed of hyperthermophiles, I believe, in hyperthermophilic environments. So this is corroborative evidence showing us not only that from the genomic level we can tell that they can kind of survive in the same environment, but also in the real world we see this actually occurring. For group 3, I believe there were 11 co-occurrences, and group 4, there were 4, so good across all of our different groups. And then we sort of have more new putative groups that we're adding in as we're sort of refining our signatures. So I'm hoping to get even more co-occurrences with sort of in tandem with that clustering, which is really great evidence for us. All right, to address some limitations in our study, the AI model really is only limited by the quality and completeness of the genome assemblies. Some of them are working with scaffolds, some of them aren't really complete, so getting those complete, full, high-quality genomes would be great. And then also verifying the accuracy of environmental labeling. Some of them we kind of only have broad parameters, but it'd be really good to narrow down on the specific temperatures and pH that they're adapted to. We can also improve our modeling by modifying different algorithms and testing out our software on different VMs to sort of enhance that computational speed. With our output, we're hoping that with those improvements, we can have improved accuracy and have a greater resolution of the genomic signature and fine-tune it even more. Future data is going to explore radiation, salinity, and high-pressure environments, as well as a couple of parameters, where a preliminary investigation of radiation, which I actually presented at last year's EMGS meeting, showed in fact that there is a radiation-resistant signature across the genomes in bacteria and archaea compared to non-radiation adapted species. Additionally, in the future, genome selections could include organisms beyond prokaryotes, so including eukaryotes as well, which would be great to kind of see if that environmental signature is shared across domains, all 3 domains, as it is with bacteria and archaea. Right, so with that, I'd like to thank my supervisor, Dr. Kathleen Hill, as well as all my collaborators, funding resources, and thank you for listening.
Thanks so much, Joseph. That was great. Really interesting stuff. I think for the sake of time, we have time for one question, but we'll move on after one. Does anybody have a question? Yeah.
Maybe just a short question.
Yeah.
Are there any plans to compare the genomic signature with a transcriptomics signature?
Right, yeah, we haven't started with that yet, but with transcriptomics, it's cool because you can take those RNA-seq reads and stuff and then kind of stack them up, so what we're calling a stacked CGR. So we just have to kind of tweak that software a little bit where instead of having a genome where it's kind of end-to-end, we have to find a way to connect those different transcriptomic reads, but that's definitely something we're interested in, very hopeful to do in the future.
Yeah, lots of cool follow-ups there. That's great.
Thank you.
Okay, thank you.